I'll talk about induced pluripotent stem cell models of human neurodevelopmental disorders. And unfortunately, it's hard for me to see on the screen here. All right, I will try and look across at this tight angle here. I know, I can move across and speak like this. Right. So we use induced pluripotent stem cells and for disease modeling and potentially for drug discovery. And the idea is on the left, if you take an individual or a patient for any type of disease, you can reprogram those skin cells that you can take from that individual and make them into stem cells that resemble embryonic stem cells. And so these embryonic or iPS cells, as they're called, can be differentiated into specialized cell types of any interest. So for neurodevelopmental disorders, we would want to differentiate them into neurons, into brain cells. So you, in the middle, you show the specialized differentiated cells, which in our case here would be neurons. And up above, where it says A for disease modeling, you can grow these neurons in a dish and try and determine what may be different between the disorder neurons and the unaffected neurons. If you can find some phenotypes, some differences between these cell types in the dish, you may be able to proceed to use them for drug screening or drug discovery and potentially for toxicity tests. And so that's where, in the end, you may be able to come towards new drugs and bring them back to the patient. But really, the starting point is to be able to make the induced pluripotent stem cells, make neurons, and try and find, are there any differences? And so how do you make induced pluripotent stem cells? What's shown on the left here is skin cells that can be taken, can be grown from a small skin biopsy. And you deliver 4 genes into these skin cells. These genes normally control growth of embryonic stem cells, so very early embryo cell types. But you're not working with any embryos, you're not destroying any embryos, you're working with skin from an individual. And after about 4 weeks, iPS cell colonies come up. So on the right-hand side where you see this big circle, that's a colony of iPS cells. You can then pick those and expand them and direct them to differentiate into essentially any cell type of the body. So if one is interested in a cardiovascular disease, you can make vascular cells, you can make hepatocytes, which would be liver cells, and you can also make brain cells or neurons. So in our case, of course, we're interested in working with neurons. How can you differentiate the induced pluripotent stem cells into neurons? What you really want to do is recapitulate normal human neurodevelopment, where you're starting off from early embryo cells, which would be on the left there, the iPS cells that we've just made from the individual who is either affected or unaffected. You can then grow them as little spheres, so that's what's described there as embryoid bodies. So that's a cluster of cells growing as a sphere. You then plate them down and they adhere onto the plastic surface of a tissue culture dish and grow as neural rosettes. Then manually a technician would cut out these neural rosettes and expand them, and they start growing as neural progenitor cells or NPCs over on the right-hand side. Now these neural progenitor cells, if you grow them in the right conditions, can differentiate into neurons, and you can get a plate of neurons that would come from that individual who has ASD or an unaffected control. Now the best way of evaluating the neurons that you make from the NPCs is to plate them out on astrocytes, which is shown in that bottom left. It's hard to read the text, but those would be astrocytes that you plate the NPCs on. And then after, it can take up to 3 months for the NPCs to differentiate into neurons and for those neurons to start to connect and grow with each other, which is shown over here. In blue, those are dendrites that are the processes coming out of neurons. And so now you've been able to make ASD-specific neurons from an individual, affected individual. So I'm not actually a neuroscientist. I'm more of a stem cell biologist, but as was mentioned before, collaboration is critical for advancing science, and so we've been working with, well, we've been learning neuroscience and collaborating with others for, well, at least since 2008 on these stem cells work. So neurons communicate by structures called synapses, and you can see on the left there where it says neurons, the dark blob in the center is the soma. That's the central part of the neuron where the nucleus is and all the DNA. You can see all these processes coming out that are dendrites, and there's actually an axon down on the bottom part that's thinner and longer. And so synapses communicate between these axons and dendrites. From the axon, it releases neurotransmitters that are received in the dendrite. And so this connection between the two is called a synapse, and that allows the neurons to communicate, and those chemical signals that are transferred elicit electrical responses, which is how our nerves function. So how can you visualize synapses? What's shown on the left is that you can label proteins that are involved in on either side of the synapse. So proteins that are involved in the release of the neurotransmitters and on the receptor part. And so on the right-hand side here, the dendrites are labeled in blue, the axon terminals are in red, and the postsynaptic density on the dendrites are in green. And so you can see down here where the arrows indicate the synapses, and you can count up how many synapses you have. And so that's the connection between different dendrites. Now, what are the consequences of neurodevelopmental disorder-related mutations for human neuronal connectivity? We had a brief mention of some of the genes, and in fact, some of the genes that were mentioned in the earlier presentation are ones that we're studying here. So really, connectivity between neurons is a function of dendrite length and complexity and the synapse density. So you could imagine that on the left, there's that arbor, the tree of these dendrites, and all the blue dots would be the synapses. If those are the unaffected individuals, you can count up how many synapses and the length of those dendrites and compare them to the affected individual on the right, and you would see that that affected individual has an underconnectivity. There's fewer, the dendrites are smaller and fewer synapses. But one could also imagine the opposite, where the unaffected individual might be on the right, that might be the normal amount, and in some circumstances you could have overconnectivity, and it might be the ASD or neurodevelopmental disorder individual would be on the left. So over or under connectivity can be a problem, and you can start mapping it out by looking at the shape of the neurons and their trees and the numbers of the synapses. And you can go on to look at the electrical conductance as well. So I'll start with a couple of examples. I'm not going to go into the genes or their expected functions, but just how do we assess the neuronal shape and their function. So I'll start with Rett syndrome. And what we found with Rett syndrome, this paper was published earlier this year, is that they have reduced dendrites and complexity. You can see in the upper panels there, a neuron is shown in green. It's being marked with a green fluorescent protein gene. So that one neuron is in green, and other neurons are stained with MAP2 behind it. And then you can trace out the length of these dendrites and their complexity. And that's compared to the Rett syndrome neuron on the right-hand side. Now, with all of these bar graphs, which I know will be impossible for you to read, the first one on the left looks at the length of the dendrites. And you can see that the green bars show that the unaffected dendrites are longer than the affected Rett syndrome ones. In the next panel, it looks at the number of tips. So you can count up the number of tips on the dendrites in those images above. And again, you can see there are differences. The Rett syndrome ones, there are fewer tips. You can look at the complexity by the branching sort of numbers in the third bar graph. And again, those go down in Rett syndrome, and that's what's shown in the bar graph on the right-hand side is the complexity also of how many of these dendrites they are and how often they branch. So in Rett syndrome, it looks like they're immature and they don't fully form the expected branching. Now, what about measuring synaptic connectivity? You can do this with an electrophysiologist. I'm not an electrophysiologist. We collaborate with Mike Salter's lab at Sick Kids. And this is an example or a little bit of a background on electrophysiology. Eric Kandel says that electrical signaling represents the language of the mind. Electrophysiology lets us listen to the neurons. So in the bottom left, you can see a recording electrode touching a neuron there, and this is a potential readout of some of the excitatory signals that are being transmitted into this neuron. And one of them, at the bottom line of the text there, describes miniature excitatory postsynaptic currents, which each one of these bars that stick down is one of these EPSCs that is actually the release of a neurotransmitter. So when we do this on Rett syndrome with Mike Salter, what we find is that those neurons are immature. They have fewer action potentials, which are the real signals from one neuron to another, and they have reduced calcium inward currents as it's described. So the upper bar graph talks about capacity. This is essentially the amount of electricity stored in that neuron. And the Rett syndrome ones in the black bar have less capacitance. And that's expected because they're smaller. Down below the graph shows the number of action potentials, and you can see the Rett syndrome ones with the black circles in it, there are fewer action potentials. And in the center one, it starts looking at specific ions going in and out of the neuron, and you can see as it dips down, there's a little asterisk there. That's where there's less calcium inward currents. You can see that there's a difference there. Now, we know that this isn't what happens in all neurodevelopmental disorders because we've also worked with Williams-Beuren syndrome neurons and given them to Mike Salter for electrophysiology. And they also have defects in action potentials, but in this case, it's potassium outward currents that are different. You can see the bar is extending up to the right here in that panel called E with the double stars on it. There's the difference there in the outward currents. So different neurodevelopmental disorders can have different electrical outcomes. This is the mini EPSCs, so each of these bars indicates a release of neurotransmitters, and again you can see some differences between the wall types and the Rett syndrome neurons, most commonly in the frequency over in this bottom right-hand graph with the black bar showing it's very, very different from the unaffected controls. What about ASD? So we've been working with Steve Shearer, and some families have provided skin biopsies. So my lab reprogrammed them into induced pluripotent stem cells. We made neurons. We started to look at some of the shape differences. But the most prominent differences are these electrophysiological ones. So again, there's this is a family with, actually two families with mutations in patch D1. And we see decreased mini EPSCs in this. Essentially, you can see in these bar graph on the right-hand side, proband 1 and proband 2 are much reduced in comparison to the control. And so there appears to be a decreased frequency of synaptic events suggesting reduced synapse formation. But there's another family that provided skin biopsies with mutations in SHANK2, and it turns out SHANK2 and patch D1 were both introduced in the genetic talk earlier. So in this case, the frequencies actually increase in these ASD neurons. And what's shown in that colorful neurons shown there, the two green ones and the two red ones, we've marked them with genes that express a green fluorescent protein or a red fluorescent protein. And so the red ones are actually the unaffected neurons, and the green ones are the neurons from the ASD individual. And again, these are mini EPSCs. They're done in a kind of technically different way. But in this case, you can see more bars sticking down for the green cells than for the unaffected cells. And that's shown here, where the R841X is the SHANK2 ASD individuals, and you can see that in this case, that EPSC frequency is way up. So why would it be way up? And this is where we can come back to looking at their neuronal shape. So the ASD SHANK2 neurons have actually more complex dendrite arbors. I showed you the Rett Syndrome ones in the same assay had less complex arbors. And what you do here is it's hard to see the gray neuron in those concentric circles, the targets there. But as you measure each of the dendrites coming out beyond those bullseyes, you can see in this shoal analysis, as it's called on the right, that there's a peak early on. There's a lot more of the dendrites that are getting to at least to the closer circles. And so this shows that they're more complex. And then you can go back and count up the synapse numbers by doing those costains to count the synapses. You can see the SHANK2 neurons have more synapses per neuron. The dendrites are actually longer, and so they're actually over-connected. So what we've seen is the Rett syndrome and the PATCH-D1s are probably under-connected, and in this case, these SHANK2 neurons seem to be over-connected. So I'll just wrap up with a couple of conclusion statements and some future directions. Human iPS cells allow production of neurons from individuals with neurodevelopmental disorders. We've published the Rett syndrome work. There are a number of other publications on this, very little so far on non-syndromic autism. The neurons that get made this way are different from unaffected controls in their maturity based on shape and electrophysiology. And we think that the neurodevelopmental disorders show a range from underconnectivity to overconnectivity. So where are we going now? Well, our future directions is, with the Rett syndrome work, we have characterized them sufficiently that we're gearing up to try and perform drug library screens to correct some of the shape defects. And this is actually funded by OBI with the Structural Genomics Consortium as well involved, and they're providing us with some compounds to test for Rett syndrome work. For modeling autism spectrum disorders, we're using iPS cells derived from individuals with mutations in known genes such as PATCH-D1 and SHANK2. We're trying to take that work all the way through that so that we can try and publish that. Some additional directions that we're working on is, rather than going all the way back to induced pluripotent stem cells like an early embryo, you might be able to directly convert skin into the neural progenitor cells. So instead of going all the way back to an embryo and then reconstituting neural development, you can just go to neural progenitor cells and make neurons. Steve Shearer's group is working with us to generate iPS cells with mutations in specific ASD-associated genes. There are now very directed ways to introduce specific mutations into known genes in the iPS cells, or even to correct genes in the iPS cells and look to see what's the outcome of that through these shape assays and electrophysiology assays. And so that's what we want to do along with Steve and with the iPS cells that we've already reprogrammed from some of the skin biopsies, is to determine the role of these genes in neuronal shape and connectivity. So just to wrap up on who did the work, the iPS cell and neuron production is what's being funded by POND. So Weiwei and Asley make the neurons from the iPS cells. Joel and Kirill in my lab on the right have been doing the analysis on the PATCH-D1 and SHANK2, but that's all separately funded. And Steve Shearer's lab is involved with Rosanna and John, providing some of the samples and creating the knockouts. And Mike Salter and with Wenbo Zhang have done the electrophysiology, and Lucy and Seema's lab are involved in the Williams-Buren syndrome work. And I'll stop there, and thank you for your attention.